covered the amino acid derived compounds, enzymes of bioregulator synthesis and degradation, the neurotransmitters that we talked about, dopamine, epinephrine, norepinephrine, melanin, serotonin, melatonin, histamine, GABA. Melanin is the skin pigment. It's not a neurotransmitter, but one of the compounds that is derived from tyrosine. Creatine, phosphate, creatinine. You really need to know the difference between the two. Glutathione, nitric oxide, tissue-specific nitric oxide synthesis, and then some of the clinical correlations like circadian rhythm, Parkinson's disease, depression, pheochromocytoma, Huntington's disease, albinism, effect of nitric oxide. Okay. Now, for the tyrosine-derived compounds,
So,
Joe, that after the
قال ايه؟
hypothalamus, adrenal medulla, that's the region where dopamine is synthesized, and we already know dopamine is one of the neurotransmitter. And then tyrosine is also converted into epinephrine, and that happens in the adrenal medulla. Remember the inner core region of the adrenal medulla in the main cross section. The inner core makes the catecholamines. And then in thyroid gland, tyrosine is the precursor for making thyroxine. Thyroxine is one of the hormones that controls the iron and various function, fatty acid synthesis, so on and so forth. That Dr. Block covered the functions of thyroxine. Right? Okay, skin tissue makes the melanin pigment, and tyrosine is the precursor. Peripheral nerve that makes the norepinephrine. Okay, then we went into the details of the catecholamines. Tyrosine is the precursor. We talked about the tyrosine hydroxylase. It uses molecular oxygen and tetrahydrobiopterin. The product is DOPA. L-DOPA is then converted by decarboxylation into dopamine. Dopamine is hydroxylated to form norepinephrine. And then norepinephrine is methylated by the methyltransferase, which uses SAM as the methyl group donor. Anytime you want to methylate something, SAM, that's the S-adenylyl methionine, is the universal methyl group donor. Here, you are transferring the methyl group onto norepinephrine, and the product is epinephrine. And all of these compounds are very important. Okay, now in the way of clearing some of these catecholamines, the two enzymes that we talked about are catecholamine O-methyltransferase, COMT is the abbreviation. You do have to know the full name. That's the catecholamine O-methyltransferase, and then monoamine oxidase, MAO. Clinically, pharmacologically, these two enzymes are very important. There are pharmacological compounds that are targeted towards this enzyme in controlling the process of degradation, right? So the idea is to keep the compound much more longer if it is a brain tissue. Okay, so the degradative products, dopamine normally forms the homovanillic acid, epinephrine, norepinephrine. The inactive compound that you get out of the epinephrine, norepinephrine is vanillic acid. Functions of catecholamines. It prepares the body for the fight or flight response, and then increased glycogen degradation is the biochemical process, and then the increased triglyceride hydrolysis or release of free fatty acid, right? And these are preparing your body for a stressful response. Okay, and then tryptophan-derived compounds. Tryptophan is the indole-containing compound. And you see the indole ring structure there. Yes. Okay, so the product is serotonin. And that serotonin itself is a neurotransmitter. Some of the brain chromaffin cells can make serotonin, and we have looked at the functions as well. Serotonin is also called by hydroxytryptamine. And it's also made in the enterochromaffin cells, gut cells, basically, and gut feeling, all of that. That's another story altogether. But serotonin is the product Serotonin is 5-hydroxytryptamine. It's a neurotransmitter, and the further conversion of serotonin into melatonin happens only in a tiny little gland called pineal gland. It's in the brain. Melatonin controls the diurnal cycle, the light-dark cycle, and circadian rhythm, basically. And the compound is melatonin, right? You really have to know that. Synthesis of serotonin, once again, tryptophan hydroxylase makes the 5-hydroxytryptophan, and then decarboxylase will remove the carboxyl. So the theme here is decarboxylation. In all of these bioamine synthesis, you keep the amino group in the compound, but you remove the carboxyl group. The product is the serotonin. Serotonin regulates the blood pressure and the temperature, and also it's a powerful vasoconstrictor and stimulator of smooth muscle contraction. And there are several other functions, but these are the main functions that we look at. And synthesis of melatonin would involve the key steps of converting serotonin into N-acetyl serotonin. NAT is the enzyme, N-acetyl transferase. And this enzyme is highly regulated based on night or day. And then once you have the N-acetyl serotonin, it's methylated to form melatonin. Once again, melatonin controls the diurnal cycle or circadian rhythm. In the histamine synthesis, from histidine, that's the imidazole containing compound amino acid. This is the imidazole ring structure here. And it is decarboxylated to form histamine. Histamine, we know that it's a compound that you form during allergic reactions, right? And the mast cells, one of the immune cells, would degranulate and release the histamine, and there are antihistamine compounds that you take to control the histamine release, right? So very important. Functions of histamine depends on which tissue you are talking about. It mediates the allergic and inflammatory reaction. That's a pretty general response here. And then powerful vasodilator. As part of the allergic reaction, there's this flushing happens, the dilation of vessels happen, and then it also causes the constriction of bronchiole. You get this icky feeling due to an allergen. And then also the secretion of hydrochloric acid in these top here, some of these functions of histamine. Gamma-aminobutyric acid, that's one of the neurotransmitter, and we classified the neurotransmitters into two types, excitatory and inhibitory. Some of these inhibitory neurotransmitters, they keep the neurons quiet or calm. In other words, there's not much of an excitation happening for a prolonged period of time, right? And these are required. And so these are inhibitory neurotransmitters. GABA is the compound name. Glutamate is the precursor. The enzyme is glutamate decarboxylase, which would remove the carboxyl. You would form gamma aminobutyric acid. And GABA is produced in substantia nigra. So GABAergic neurons. Here, okay. And the lack of GABA production would lead to epileptic seizures in some individuals. Severity can vary. And talking about the dopamine, those are the ones that are synthesized in substantia nigra. And one of the treatment for Parkinson's disease is L-dopa, right? So the degeneration of the substantia nigra will result in Parkinson's disease, and these individuals cannot make enough dopamine. And one of the compounds that's administered is L-dopa. L-dopa is a compound that can cross the blood-brain barrier. One of the challenges with the treatment of brain disease is to make sure that you have enough drug in the CNS, right? And so you would want to have a compound that could cross the blood-brain barrier. L-dopa can do that, and so that's that. Depression, serotonin metabolism is basically affected. Serotonin in general makes you happy. In some individual, they cannot make enough of it. They undergo depression, and even a normal individual, one time or the other, depending on the situation, can go through this. And there are inhibitors, serotonin reuptake inhibitors. Prozac is the chemical Melanin synthesis, all the way from tyrosine to melanin. The enzyme is tyrosinase. It's a copper-containing, copper-requiring enzyme that uses molecular oxygen, dopamine and melanin. That all happens non-enzymatically, and skin is the issue. Albinism, tyrosinase deficiency, the skin is susceptible to UV rays, and it can lead to skin cancer. Creatine phosphate synthesis, arginine and glycine are the two amino acids that are required for making creatine. Basically form one amino acetate and eventually form creatine. Creatine is the active compound that receives phospholipid from ATP to form creatine phosphate. Right? And once you form creatine phosphate, it can be stored in tissues like skeletal muscle, cardiac muscle, and brain. And then time in need, you can remove the phosphate group and then add that to ADP to form ATP, right? So very important, this pathway. Creatine phosphate function, it serves as an energy reserve, basically for brain and muscle, and two types of muscle, skeletal and cardiac muscle. And, we won't get into minor tissues like kidney, but there are three major tissues: brain, skeletal muscle, cardiac muscle, right? Okay. And creatine kinase can serve as a clinical marker, but with the question mark, you can be fooled for heart attack, but you might be having some muscle damage due to exercise or something. So you have to watch out. Troponin T is a good marker for cardiac specific thing. So troponin T. Creatinine synthesis, basically, if somebody is having a muscle damage, the levels of creatinine is going to be higher, and the conversion, the formation of creatinine, the 9-ending, right, happens non-enzymatically. So basically, instead of transferring the phosphate onto ADP to form ATP, these individuals would form creatinine, and the levels of creatinine would be high in those individuals who have damaged skeletal muscles, for example, right? Okay, so very important. Okay, creatinine is a waste product, it's not useful, and you can measure that in the blood, and under pathological conditions they go up, but normally it's 1.5 milligrams per deciliter and in the plasma, but in the urine it's much more higher, 1 to 1.6 grams. Okay, glutathione is a tripeptide that controls the cell's redox status, and the tripeptide has the gamma-glucose plot. It's the cysteine that's the business end of the whole compound, right? Okay. Glutathione usually it maintains the protein thiol groups in their reduced state. Intracellularly, you need to keep the environment in a reduced state. Extracellular condition is different, it's mostly oxidizing. But anyhow, liver can also use glutathione as a functional group to be added on to xenobiotics. So it's a detoxification mechanism where you would transfer the glutathione group onto these compounds and you can clear that up, and liver does that. Nitric oxide is a gaseous molecule and it has several functions. The precursor for making nitric oxide is arginine, oxygen, and NADH is a reducing agent. Powerful vasodilator and a neurotransmitter, and there are three types of NOs that we discussed. Endothelium-derived relaxes smooth muscle. Brain-derived, not much is known, but activation of protein, some of the proteins. And macrophage, it functions as a weapon to kill the pathogenic bacteria. Macrophages, once they engulf the pathogenic bacterium, in order to get rid of the engulfed bacterium, you would basically use nitric oxide to kill the bacteria, pathogenic bacteria. Very important. Amino acid metabolism we continued on. Once again, this is the neuron structure, cell body, nucleus, myelin sheath that protects the axon, and the myelin, if it is not produced in proper quantities, it can lead to clinical conditions like multiple sclerosis. Glial cells, the three glial cells that we talked about: microglia, oligodendrocyte, and then astrocytes. You really have to know the functions of it. And once again, microglia is brain's own immune cells, and then oligodendrocyte makes the myelin, and the astrocytes, they are thought to clear some of these neurotransmitters, right? Excitatory versus inhibitory. Excitatory, in general, acetylcholine, aspartate, dopamine, histamine, norepinephrine, epinephrine, glutamate, serotonin. And then the inhibitory types that we talked about are glycine, taurine, and 4-aminobutyrate or GABA, right? Very important. And stages in neurotransmission, we basically introduced the concept of neurotransmission. You have presynaptic neuron and postsynaptic neuron and the resting state. We chose the example of acetylcholine. It's basically bound to or stored in synaptic vesicle, and due to environmental some sort of a stimulus, like for example the influx of calcium, the acetylcholine vesicle will fuse to the postsynaptic neuron or presynaptic neuron, and it will release the acetylcholine, which would bind to the postsynaptic neuron, right? The receptors that are located in the postsynaptic neuron. And then the termination is basically degrading the acetylcholine, right? Glial cells, once again, oligodendrocytes, they make their fatty myelin sheet, and they basically insulate the axon, myelin. And if you don't make enough of it, you would end up with a condition called multiple sclerosis. There's movement imbalance, and then disruption of physical movement, and also the vision imbalance. GABA in epilepsy. If you don't make enough GABA, you end up with seizures, epileptic seizures. Some of the compounds that are available for treatments are phenobarbital, benzodiazepam, Keppra, Depakote, and so on. GABA normally degraded by this pathway here. GABA is converted all the way to succinate, the TCA cycle intermediate, right? And the intermediate here is succinate semialdehyde, right? There's transamination involved and then dehydrogenation involved. The point here is GABA is the inhibitory neurotransmitter, and succinate is the product that cannot do inhibitory function. Okay, now role of acetyl-CoA. Acetyl-CoA is a C2 activated unit that can be used in many pathways. It's involved in TCA cycle. If you want to acetylate chromatin, you can remove the acetyl group from acetyl-CoA, and you can modify histone proteins, right? And you can regulate the gene expression based on acetylation, deacetylation status, right? Okay, isoprenoids. As part of the cholesterol synthesis, we looked at farnesyl and geranol. Those are isoprene units that can be used for modifying oncogenic proteins, right? And then acetyl-CoA is also the sole precursor of C2 unit that's required for fatty acid synthesis, cholesterol synthesis, and then also the neurotransmitter acetylcholine synthesis. It's very important. The making of acetylcholine would involve the use of choline as a substrate, and the enzyme is choline acetyltransferase. You would basically add the acetylcholine onto the hydroxyl to form acetylcholine. Acetylcholine itself is a neurotransmitter, very important. And we talked about the cholinesterase enzyme, one of the fastest enzymes that would degrade the acetylcholine, and you are expected to have that type of a fast reaction. When you're talking about neuronal function, the action potential lasts for a few seconds, like 10 seconds, and then you want to turn off the system, and you need the enzyme cholinesterase, right? Okay, now acetylcholine basically opens up the sodium potassium channel. That's physiology. You'll get that in the cell, of course. Okay, physostigmine, neostigmine, those are the inhibitors of the acetylcholinesterase. It basically carbonulates the sealing residue of the enzyme and basically inactivates the acetylcholinesterase. And then there's also DIPF, that's one of the organophosphonate nerve gas poison that modifies the acetylcholinesterase and completely inactivates it. And so that's that. DIPF, that's the structure here, and phosphate would have to have 5 bonds, oxygen moving two double bonds with the oxygen and one more. So pentavalent here, and there's isopropyl here, isopropyl here, and so diisopropyl fluorophosphate, right? Very important. Okay, and then here's how the enzyme gets modified. Fluorine is a good leaving group. The serine oxyanion will react to dephosphonate to form the inactive adduct, right? So the enzyme is permanently inactivated. It's an irreversible type of inhibition that we talked about earlier. Okay, now acetylcholine receptor targeted inhibitors, some of the venoms like cobra toxin, alpha bandora toxin, tuberculin, all that, they bind to the acetylcholine receptors and then does the damage. Proline synthesis, glutamate is the precursor, proline is the product. Proline gets modified, get the hydroxyproline and vitamin C independent reaction. We talked about that. Selenium is one of the trace elements that's required for normal functioning, and serial tRNA is converted into selenocysteine tRNA. And one of the enzymes that has selenocysteine is glutathione peroxidase. Dr. Grosskopf will be getting to that. Then she talks about the oxidative damage. There's one protein, glutathione peroxidase, that has a few residues of selenocysteine, and role of selenium is very important in terms of making sure the redox status of the cell is maintained, right? Once again, you want to keep the cells in the reduced state, intercellularly speaking. And if you have oxidative damaging conditions like oxygen radical, that can kill the cell. So anyhow, selenium is important. Tyrosine hydroxylase that takes the phenylalanine and converts that into tyrosine. Lack of it would lead to a condition called PKU, phenylketonuria, very important. Autosomal recessive mode of inheritance. Alkaptonuria, basically the oxidase is missing, and you would accumulate the product homogentisate. Homogentisate can form this polymeric compound, accumulates in joints and causes osteoarthritis. SAM synthesis, methionine is the amino acid, sulfur amino acid, and then you would add the adenylyl group to form the SAM, and it is this methyl group that gets transferred onto various recipient compounds, and you get the methylated product. DNA methylated product, RNA can be methylated, so many compounds. Okay, so here are the list of compounds that are methylated products here. Creatine phosphate, phosphatidylcholine, epinephrine, methylated RNA, trimethyl lysine, and then histones can be methylated. It's a big thing now that epigenetic mode of controlling gene expression is through methylation, demethylation. That's one mechanism, right? And then CPG islands, DNA has this CPG islands that can be methylated, and you can control the gene expression. So very important methylation. Yes, quick.
During lecture, like,
But we didn't talk about this. You have the slides for it.
yeah, but like I asked you, and you said that since we didn't cover it in class, we weren't responsible for anything like past.
These slides you have to study. Okay. And then the post-translational modification, N-acetylation, N-methylation, ubiquitination, O-phosphorylation. And histone is a good example. Histone gets methylated, histone gets ubiquitinated. Histones can also be phosphorylated. And then for acetylation, methionine and serine gets acetylated. Here's the acetylation of protein. It changes the whole chromatin morphology, and it changes the whole topography of the chromatin, and you control the expression of the gene at the epi level, right? Very important. Okay, we won't get to that. Cysteine synthesis, methionine is the precursor, and eventually you form cysteine. There's two clinical conditions that we looked at. Cis-ethylene beta synthase. which is cystathionine gamma diase or cystathionase. These two steps are important. If you have a block in here, you would have homocystinuria, which means you are accumulating homocysteine. But if you have a block over here, the product that is mostly accumulated is this one here, and that's cystathionuria. Between these two compounds, this is more deleterious, and we have already looked at homocysteine in depth, right? Homocysteine can form homocysteine dilactone, it can modify LDL, and so on and so forth. And this is very important here. Homocysteine is the substrate, product is methionine, the enzyme is methionine synthase. Vitamin B12 is required, also folic acid is required, and you will basically transfer the methyl group onto the homocysteine to form methionine, and the lack of it, once again, would lead to homocysteine accumulation, right? So here are the sulfur amino acids. Cysteine, cysteine is the oxidized form, mind you, that's the oxidized form, that's the reduced form, cysteine. So cysteine has this thiol group, but cysteine has the disulfide linkage. And homocysteine and versus homocysteine. Homocysteine has the free thiol group. This homocysteine is the disulfide, right? And in terms of the solubility, these two compounds are insoluble. They can aggregate, precipitate, and do damage, right? Okay. Cystinuria is the other condition that we talked about. That's the defect in the dietary transport of the cysteine. Okay, mixed disulfide tryptophan is degraded. The useful products are nicotinamide monophosphate and then formic acid, that's one carbon. Branch chain amino acid degradation is a condition that we looked at as the maple syrup urine disease, MSUD. And this enzyme is defective, and we really looked at that. Thiamine pyrophosphate, lipoic acid, pantothenate, vitamin B, vitamin B2, vitamin B3, all goodies are required for that enzyme, right? And this part here you really have to memorize. Okay, and then we talked about propionyl-CoA metabolism, where propionyl-CoA is converted into methyl-malonyl-CoA and then succinyl-CoA. And then this connection here, methyl-malonic aciduria. And then histidinemia, where you lack the enzyme histidase and you cannot make uracanate, and that's a problem if it is a skin tissue, also downstream. Okay. Carnitine is the compound that is usually added on to the fatty acid, and you require that for beta oxidation. Lysine is the precursor. Lysine can be categorized all the way to acetoacetyl-CoA, eventually acetyl-CoA, and it's one of the pure ketogenic compounds, right? Leucine and lysine. Phosphocreatine, you require lysine arginine, tetrahydrofolate, you really have to focus on the folic acid. The business end of the compound is N5 nitrogen and N10 nitrogen. This is where the one carbon is added. You have N5 methyl tetrahydrofolate, N5 N10 methylene tetrahydrofolate. So many varieties of folic acid you can charge on either between N5 and N10, or individually with N5 or individually with N10, right? N10 formyl tetrahydrofolate is required for purine synthesis, for example. But if you dissect the compound, you have pyridine ring, you have the TABA portion, and the polyflutamate portion. But the business end of the compound is between it's N5 and the N10, right? Folate, that's the vitamin B9, can be converted all the way to tetrahydrofolate. Bacteria can start from GABA and forms the folic acid. That part is bacteria specific or prokaryotic specific, and there are sulfur drugs that can inhibit that stuff. And then you also have this step that can be inhibited, that's the dihydrofolate reductase step that will inhibit the conversion of DHF to tetrahydrofolate, right? Very important. Okay, folate metabolism one more time. Vitamin is the B9 vitamin. folic acid, and here's the butane mechanism. Basically, you're charging one carbon onto the folic acid, and then you're stripping the one carbon for purine pyrimidine use, for example, right? During the course of purine de novo synthesis, you would strip off the one carbon and use it for that purpose, right? So you really have to focus on how one carbons are charged. And here's again, one more time, homocysteine to methionine conversion. You would strip off the methyl group to form the methionine. And once again, this is the vitamin B12 step. Right? Okay, and this is a nice summary of where one carbon, for example, it comes from tryptophan, and you use it for purine synthesis. Histidine donates the methionine group, you use it for purine synthesis. Serine glycine, they donate the methylene group, you use it for thymine synthesis. And then you would use the methyl group to form the methionine. Megaloblastic anemia, we really talked about the lobes here. Normally it's trilobe, 1, 2, 3. And if it is pentalobed, that's megaloblastic anemia. Basically, the neutrophils are not dividing, it's stuck in that form. You have 5 lobes in there, and all because of the folate trap. Right? So folate trap here, you're trapping the folate in the form of methyl. You're not making thymidine here, and so the cells are not dividing. So very important pathway here. Serine glycine synthesis. You form glycine from the 3-phosphoglycerate, and also you can strip off the methylene group and then add back to THF. Glycine can be used for serine synthesis, heme synthesis. Remember, one of the amino acids that's required for heme synthesis is glycine, right? Succinyl-CoA plus glycine. And then glycine is also required for purine synthesis. And glycine is also one carbon donor that can donate its own carbon that can be charged on folic acid. So it's very important. Taurine, and then this is a summary of all the diseases, where the block is, and then what type of a disease you would manifest. For example, cystathionine beta synthase, homocystinuria, methylmalonic aciduria, branching ketoacid dehydrogenase defect, that's food. And then you also have the homogeneous oxidase, that's the alkaptonuria, and then phenylketonuria, right? Okay. Purine pyrimidines. Purine is bicyclic. Pyrimidine is monocyclic, and the two purines are adenine and guanine, AG, and cut pi, right? So, cytosine, uracil, and thymine. Those are the pyrimidines, monocyclic, and you can see that methylated thymine is methylated cytosine is basically thymine. Okay, and then ribose versus 2' deoxyribose. You really have to know this. Ribose has a 2' hydroxyl, but then deoxyribose, it doesn't have a 2' hydroxyl, right? And the difference between DNA and RNA fundamentally, you know that. In adenosine cytidine, you can see purine that is attached to the ribose, right? Because that's a nucleoside. And here's the pyrimidine nucleoside. And then the salvage pathway versus the degradation of purine. So if you have ribose 5-phosphate, that's the sugar phosphate that you need. You would make PRPP, and PRPP is the phosphoribosyl donor, and that's used in salvage pathway as well as the de novo pathway. And then you can make nucleotides, and once you have the nucleotides, you can do RNA, DNA synthesis, right? Okay, so from PRPP, that's PRPP synthesis, PRPP synthetase, and PRPP is converted into 5'-phosphoribosylamine, and once you have phosphoribosylamine, you can construct the purine ring on top of the phosphoribosal, right? Okay. PRPP aminotransferase is the enzyme, glutamine is the amino donor. And this enzyme is regulated, highly regulated. The PRPP, that's the substrate forward activation. And then the products like IFP, AFP, GFP, those are inhibitors, shutting the synthesis off. Okay, 5-phosphoribosylamine in 9 steps, you get the IMP, the first cyclized purine ring structure, that's the inosine or hypoxanthine base attached to the sugar. Purine skeleton one more time, and then purine skeleton, you need nitrogen from glutamine, that comes from glutamine, carbon 2 and 8 comes from forming folate, right? And then carbon 6 comes from bicarbonate, nitrogen 1 comes from aspartic acid, and then this glycine is the one which donates carbon 4 and 5 and the nitrogen, okay? Okay. Once you have IMP, it's converted into AMP in 2 steps, and aspartic acid only donates the amino group here, right? And then forms AMP, and then IMP is also converted into GMP, where you have the intermediate XMP, the enzyme that is highly regulated is the IMP dehydrogenase. Okay. So schematics of the purine nucleotide synthesis. You start out with 5-ribose 5-phosphate, you form the phosphoribosal, and phosphoribosal is the PRPP is the phosphoribosal donor, right? Whether it is salvage or de novo. Okay, then you have all these amino acids, glycine, aspartic acid, glutamine, donating either nitrogen or carbon or both. Have to know that. Then hypoxanthine is converted into GMP or AMP. Regulation of the 2 enzymes, the mineralosuccinate synthase and the ICA hydrogenase. Salvage pathway, basically using the pre-existing nitrogenous base and forming the nucleotide. So through diet, you consume or you have this nitrogenous bases which can be converted into nucleotides. And the 2 key enzymes that we talked about is the HEPRT, hypoxanthine guanine phosphoribosal transferase, and then APRT. Okay, so here's the salvage pathway. Once again, you can take hypoxanthine, convert that into IMP, guanine, GMP. Both are catalyzed by HEPRT. Adenine goes to AMP, that's the APRT. So erythrocyte cannot perform de novo, so we depend on the salvage pathway. Tetrahydrobiopterin comes from GTP, THP is used for a lot of these hydroxylases, like phenylalanine hydroxylase, tyrosine hydroxylase. Purine degradation. So here's how these are very important here: hypoxanthine to xanthin, xanthin to uric acid. Those are catalyzed by xanthin oxidase. Xanthin oxidase uses FAD, molybdenum, iron, right? One of the only enzyme that uses molybdenum as a cofactor is the xanthin oxidase. You also see purine and
Huh?
these three are clinically relevant.
Yeah.
The purine nucleoside phosphorylase or adenosine deaminase and xanthin oxidase. And then pyrimidine synthesis. Before we get to that, the 2 clinical conditions that we talked about, the gout, either increased PRPP synthetase or decreased HEPRT activity can lead to increased de novo synthesis of purines, then increased uric acid production, right? And von disease versus gout, very important here. And the treatment for gout is allopurinol, alaxanthin. Lesch-Nyhan syndrome, this is X-linked hyperuricemia. You have the lack of HEPRT protein or enzymatic activity. Once again, the end result is excess uric acid synthesis and gout. Combined immune deficiency, that's defect in adenosine deaminase, and you have a defective T cell and B cell immunity. And then you also have purine nucleoside phosphorylase deficiency, that's a T cell dysfunction. Okay? Okay, the reason why you get combined immune deficiency because of the TNT and you get lysindiamase is listed there. And one of the treatment plans is to transfuse blood, or bone marrow transplant, and perhaps gene therapy. Pyrimidine, single ring structure, glutamine, bicarbonate, aspartic acid. Those are the amino acids, and plus the one carbon bicarbonate, right? One here. Furin versus pyrimidine. Pyrimidine synthesis. Carbamide phosphate synthase 2, that's the cytosolic version. You form the cyclic compound with dihydroorotate, and then you get orotate, and then the orotic aciduria, you get the accumulation of orotate, right? That's an acidic compound. And one of the treatment plan is to use uridine. Okay, UMP goes to ADP and then eventually form CTP synthesis, that's the enzyme CTP synthase. Basically, you are adding an amino group that comes from glutamine, and it's a very interesting enzyme. Mechanistically speaking, it uses glutamine as the amino group donor. You would transfer that onto the UTP to form CTP.
سيبني لهاك بعديها.
Regulation of pyrimidine, carbamide, orotic aciduria versus uridine. Okay, so this is the cell cycle, and you want to make deoxyribose, the enzyme is ribonucleotide reductase, and hydroxyurea can inhibit the ribonucleotide reductase step. ATP activates it, deoxy-ATP inactivates the ribonucleotide reductase. So this slide, it's a very complex slide, but very important slide because you're making the deoxy version here, and it can be targeted in terms of cancer. For example, cancer cells, they would have to have the deoxyribose to make new DNA, and you can block this activity here to the hydroxyurea. Also, endogenously, you can inhibit this enzyme with deoxy-ATP and activate it with regular ATP. Okay, now TMP synthesis 5-fluorouracil inhibits the thymidylate synthase. Dihydrofolate reductase is inhibited by methotrexate. You really have to memorize all the drugs that we talked about, the functions of it. NAD is the coenzyme that comes from vitamin D3, that's niacin. FAD comes from vitamin B2, that's the riboflavin. And the coenzyme A comes from vitamin B5, that's the pantothenate. This is the 6-mercaptopurine serves as a negative effect of the PRPP aminotransferase and aminosuccinate synthase. So basically, you're stopping the de novo synthesis of the DNA. Right? Basically, you're stopping the purine de novo synthesis and then eventually the DNA synthesis. Okay, 6-mercaptopurine. 5-fluorouracil, once again, that's the inhibitor of thymidylate synthase. And it's a potent inhibitor of thymidylate synthase. That's the 5-fluorouracil. Add a C, it's arabinose instead of ribose. That's the problem with that compound. And then hydroxyurea, once again, if you look at the hydroxyurea, that inhibits this step here, the ribonucleotide reductase, that is the conversion of the ribose into deoxyribose. Right? Create any thiazofurine, that's a non-specific compound that inhibits IMP dehydrogenase. Allopurinol that inhibits the xanthan oxidase, right? So allopurinol here versus xanthan oxidase. AZT, that's the compound that inhibits the viral RNA-dependent DNA polymerase, and you see that the 3' is modified. AZT basically inhibits the DNA chain growth, right? DNA polymerization. And it's the viral RNA-dependent DNA polymerase that gets potently inhibited, 100-fold more sensitive to this enzyme. Acyclovir, that's another compound that's used for HIV control. Methotrexate, that's the inhibitor that's used for inhibiting the dihydrofolate reductase. Methotrexate has a Ki inhibitory constant of 0.1 nanomolar, pretty high bind. Methotrexate basically inhibits the TMP synthesis. Now, glutamine antagonists, these are very non-specific because glutamine as an amino acid is used for very many bodies as a nitrogen group donor. So you're inhibiting multiple factors. And so it's not a very useful compound because it's so cytotoxic. The host cell can also be affected, the normal cells. Okay. I don't have much time. Good luck with your exam.